When an action potential arrives at the nerve terminal, it triggers the release of neurotransmitter. In this video, we'll look at the anatomy of the axon terminal. We'll also look at the events that lead to neurotransmitter release. So recall that axons are these polarized cells. Action potentials are initiated at the axon initial segment. They travel down the length of the axon, where they terminate in a synapse between the presynaptic cell and the postsynaptic cell. These structures have a lot of specializations, and there are a lot of features to them. Here's a cartoon version that I've drawn. This would be our myelinated axon terminating in the axon terminal, and the action potential would travel all the way down to the end of the terminal. Axon terminals are also called axon boutons or axon knobs. You'll see those terms. Over here, I've got the postsynaptic cell. Now, often, the portion of the postsynaptic cell that receives a synapse is the spine on the dendrites. This isn't always true, but it's often true. At most synapses, there's a space between the presynaptic and the postsynaptic cell called the cleft. And if we take all of these together, the axon terminal, the postsynaptic cell, and the cleft, together these define a synapse. And really, I should say that these are a chemical synapse, because there's another type of synapse called an electrical synapse where there wouldn't be a space and they'd actually physically be in contact. But we'll leave that for another day. Let's look at these structures and try and look at the cellular specializations that are present. So here's a myelinated axon. We've seen these before. So for example, you know that in the axon of neurons, there are voltage-gated sodium and potassium channels which are generating and regenerating the action potential. The axon of neurons also contains an extensive cytoskeleton, and the types of cytoskeletal elements that are found in the axon are microtubules and neurofilaments. Now, when we get to the axon terminal, things look different. Probably the biggest difference is that we see that there are many vesicles that are present, and these vesicles are filled with neurotransmitter. There is a cytoskeleton in the axon terminal, but unlike the cytoskeleton in the axon, the cytoskeleton in the terminal is dominated by a protein called actin. There are quite a few mitochondria in the terminal. Synapses are expensive to maintain, metabolically expensive to maintain, and so we need to have a way to generate ATP. There are numerous membrane proteins present in the membrane at the terminal. Often these are lumped together into dense zones, which are called active zones. But not all proteins are present in active zones, and you'll find them other places in the membrane. One important protein that we see in the terminals are voltage-gated calcium channels. Now, we've seen voltage-gated channels in the axon, but those were voltage-gated sodium and voltage-gated potassium channels, and we have not yet seen in a neuron a voltage-gated calcium channel, but at the axon terminal they're present and they play a really important role. The postsynaptic cell has a number of specializations as well. So it also has a cytoskeleton, tends to be an actin cytoskeleton. It also is going to do a lot of metabolically expensive things, and it has mitochondria. In the case of dendritic spines, we often see an extension of the smooth ER that extends into the spines. This serves a number of functions, but one of the ones that's important is it can act as a store for intracellular calcium, so that the cytoplasmic levels of calcium are very low, but there's a source of calcium nearby. Like the presynaptic cell, there are numerous membrane proteins present in postsynaptic cells, and we can see these in electron micrographs, and we'll call them the postsynaptic density. Not all proteins are in the densities. There are also some elsewhere in the membrane as well as in the cytoplasm. Well, I want to show you what a synapse actually looks like in an electron micrograph. So this is an image of an electron micrograph showing our presynaptic cell, the axon terminal, and the postsynaptic cell, which would be the dendrite of some postsynaptic cell. And I want to show you that the structures that I've drawn in the cartoon are really present. Let's look at this presynaptic cell. Here, for example, is a mitochondria, and maybe you can see the double membrane that defines that. Here, and I haven't bothered to circle all of them, but here are some of the synaptic vesicles, which would be filled with neurotransmitter. You can see that arrayed opposite them, there are these fuzzy, dense structures. This is the postsynaptic density, and I've highlighted it here and here. There's also a presynaptic density at the active zone, which I've highlighted here, and I think you can see that the membrane there is specialized and there are a lot of proteins that are present that will be involved in transmitter release. I want to talk about the events that are going to lead to transmitter release. When an action potential arrives at the terminal, well, we know what happens. In the axon, the action potential has been a traveling wave of excitation where voltage-gated sodium channels have been opening up, allowing sodium to flow into the cell. That positivity can spread into the axon terminal where the voltage-gated calcium channels can be activated so that calcium can enter. Calcium does a number of things in the terminal, but most important for our purposes is that it can facilitate the fusion and then the release of transmitter from these vesicles that are present in the presynaptic cell. That neurotransmitter then can be received by the postsynaptic cell, and that's how we're going to pass along a signal. I want to talk about the specific processes that are involved in the fusion of synaptic vesicles, because it turns out that this isn't really a trivial thing for the cell to do, and there's a whole bunch of specialized machinery that's involved. This is a big blow-up of a synaptic terminal. We're looking at the cytoplasm here, and I've just drawn a single vesicle filled with neurotransmitter, and the synaptic cleft would be the white space at the bottom of the screen. To get the membrane of the vesicle to fuse with the plasma membrane will require some helper proteins. These are called SNARES, and they come in two flavors or two broad classes, vesicular or V SNARES, and then target membrane or T SNARES. I will not draw all of them, but I will draw some of the more important ones. So for example, there's a protein called synaptobrevin, which is a vesicular SNARE. We find it in the membrane of the vesicles. Here are two examples of T SNARES or target membrane SNARES, syntaxin and SNAP25. And what needs to happen for us to get the vesicle to fuse and release a transmitter is that these have to form a complex. Another key player that we'll see will be synaptotagmin, and this protein has some calcium binding domains, and you can imagine that the role of calcium then will facilitate in the fusion. So what's going to happen? Well, there are some proteins that are involved in docking and priming the vesicles, so even before they get released, some steps pre-happen. So for example, there's a protein called MUNC18 that comes in, changes syntaxin so it can then go and associate with synaptobrevin and SNAP25. And at this point, we've got this SNARE complex, and the vesicle is docked at the membrane. However, the membranes haven't fused yet. It's not able to release the transmitter. It's just in a position that's ready to go. To get release, we're going to need to bring in calcium. So when the action potential arrives at the terminal, calcium will enter the cell through these voltage-gated calcium channels and bind to the synaptotagmin. This causes a conformational switch, and that then allows the two membranes to be pulled even closer together, allowing the release, the fusion of the membrane, and the release of the neurotransmitter. Now the transmitter will diffuse across and it can interact with the postsynaptic cell, but in the meantime, the presynaptic cell has to recycle this machinery so that it will be able to reload it. It does this with the help of even more proteins. So that proteins will come in, they'll associate with the bit of the membrane that was a vesicle, and then take that membrane and endocytose it to bring it back into the cell. And as this bit of membrane is taken away to be recycled and refilled with neurotransmitter, another synaptic vesicle can come to the active zone where it will be able to then fuse and release its contents. To recap, the arrival of an action potential at the axon terminal allows calcium to enter the cell, and that then mediates the fusion of synaptic vesicles so that transmitter is released into the cleft. This signal then will have to be detected by the target cell membranes, and what happens in that target cell will depend on the particular type of neurotransmitter that's been released and the type of neurotransmitter receptors that are present in the cell. We'll look at that in another video.